Yeah.
I told her, I was like, I'm still gonna go, cause like, it's like really big now, so I was like, but like, I'll have the gym. But I think that's why she's just like behind.
Yeah.
Like high up, nothing.
That's probably good.
I know, like that's what I told her, like these chapters, I'm a little overwhelmed, like
Yeah.
I'm like. They're gonna like. That just like not lined up, like in green, then it wasn't in there.
Yeah.
So it's like.
Yeah, it's so.
Morning.
I did not know they were gonna have.
Oh, I thought I sent an email about it. Yeah, it's gonna be all week.
So it's
Yeah.
like.
That's a national firefighter competition or something.
Especially for another
Yeah,
class.
exactly.
So it's just all, I could always just have. a check.
Yeah. And get no direction.
Yeah, but it'll be really helpful this week.
Yeah.
I'm hoping.
Okay. I don't think anybody is on Zoom.
Really?
People, I mean, you guys know there's points for. Well, but you know, we're allowed to miss, you know, I say 2, not 2 days. So 1 day is 2 exercises, right? So just to FYI. So that's okay. This is the day. This is not the day I would miss, as I'm sure I heard you guys saying. Chapter 6 is not the chapter to miss.
Okay.
Chapter 10 also next week, not a chapter to miss. 13 and 14, if I had to pick a day to miss, that would be the day, but also, you know, you're still gonna get tested on it or whatever. But like chapter 6 and chapter 10 are definitely by far the hardest chapters in this unit. And they are not the ones that I would be missing. So hey, if your friends weren't here today, maybe spread that word along, like they should probably watch the recording. Because I don't want people to miss out on that, you know, if they get something out of this stuff.
Not be able to make it simple, like log.
Yes, if you can't make it and your kid is sick and there's like verifiable evidence. So I'll say, if you're logged in online, it's equivalent of coming in person. That's fine. Yes, that's the equivalent. And if I call on you, then you guys can type in the chat. And if you call on you and you're not participating, I'll just remind myself to come back to you later on. So just keep that in mind. You can still get called on and still get your credit if you join via Zoom. If you can't join via Zoom, like your kid has a doctor's appointment or something, right? Then send me evidence of why you missed. And then usually I'll let you watch the video, or like, I'll tell you what you need to do to get your points back. But if there's a reason for missing, then you can get your points back, right? All right. Let's see. Let me just sort this. Sorry. Make sure everybody's signed in. Here.
Oh.
Yeah.
Okay.
1, 2, 3, 4, 5, 6. That's wild to me that there's 9 people here. Whatever. Okay, so we're gonna go ahead and get started. We're probably gonna have a lot of people here that you know, we're gonna miss a lot of people. So when I call on people, we're gonna be going through a list until we find names. We're still gonna go in order. All righty. First question: what is the term that refers to an organism's total sum of genetic material? Of these choices, let me move this. Actually, let me hide this. Yeah, okay. So this is going to be genome. That's the term referring to the whole sum. What some of the terms that are on here? Actually, let me see. I forget. I have to call on you guys. It's not just on me to do this. Kylie, Lippy. Yes. So why isn't it chromosome? Why is chromosome not the sole sum?
It's just part.
It is just a part of it, right? So we can have extra chromosomal content as well. So when we talk about bacteria, one of the things it talks about in the chapter is plasmids, which is one of the other choices on here. So plasmids are little extra bits of circular DNA. They're still DNA, they're just in a little circle. And we might talk about bacteria. Bacteria's genome, their chromosomes are in a circle too. So what's the real difference there? The difference is that their main genes that they need to survive are on their chromosomes. They might have extra genes that help with like antibiotic resistance or toxin production and stuff like that. They can survive without them, but those genes might help them survive better. Those are often present on plasmids. Okay. And we also know that in our own cells, in eukaryotes, we have learned that our mitochondria have their own DNA, and that's not part of our chromosomal DNA. And so, yeah, so we want to consider all of the genetic content in the cell as the genome. So excellent answer. All right, next question. All right, the physical expression of a genotype. We can also think of this as what's expressed, the traits we see in an organism. What's the term for that? Jeremy's our next person, and he is not here yet. Kaylee Jackson here. No. Jimena. No. London McNair. I don't think London. Yeah. You're London. You're the other London. Okay. Yes, I'm trying to keep track of London. Cooper isn't here, right? Yeah. So okay. You're okay. Just want to make sure that we're like, yeah. All right. So physical expression of a genotype. So obviously this isn't, I wouldn't think this would be genotype and nobody picked it because I mean, I feel like physical expression of the genotype. Obviously we're looking for a different term here. So here we have the phenotype, physical phenotype. That's one way you can think of it. What do you see physically in the organism, right? I kind of already explained it and jumped ahead. So we'll come to you next. Okay. Cool. Like there's not many people in here, so we'll have a lot more opportunity. we'll have a lot more opportunity. I have a night class on Tuesdays and Thursdays that is only 13 people in that class. And so this process is a lot easier on them. It is for you guys typically, but today you get the easy track. All right, so that's easy enough. Genotype is going to be like your actual genetic makeup. So really quickly, we get genes from our mother and from our father. You have 2 copies of every gene in your body. They are not both expressed. There's only one of them going to be expressed for each gene in your body. How does your body determine which of those genes is going to be expressed? That's based on dominance and recessiveness, which isn't covered a lot in our book. That's something that is not really as important that we learn about for that. But it is determined by dominance and recessiveness. And what that means is a dominant trait will be expressed over a recessive trait. And there's a lot of gray area, and there's some genes that you might have multiple different versions of. For example, eye color. Is there just green and brown? No, there's like a lot of different ones. And then like which genes you have, some of them might be more dominant than other ones. And there are even some cases where you can have maybe both genes expressed in some way. And even some tissues, we might have this tissue, like this eye might be one color and that eye might be another color. They're getting different just signals in those tissues for whatever reason, often due to signals that we're getting in birth. It could also be due to having slightly different genetic makeup in those tissues. If you had like what we call a parasitic twin that you absorbed, so you have tissues that are literally genetically different within the person's body. And so that stuff can all basically predict how those things will be expressed. So moving on to that next question. All right, 3 parts to a DNA nucleotide. Make sure we look at all of our choices here. All right, so the 3 parts to a DNA nucleotide. We have for our correct answer, deoxyribose sugar, phosphate, and nitrogenous base. Okay, so London, this is going to be your question here. When we're talking, what are other choice? What was popular? Deoxyribose sugar, sulfate, and nitrogenous base. Obviously, the difference is the phosphate group, right? So tell me a little bit. I'd say a little bit about how DNA structures. We have a backbone of the ladder, and we have things in the middle. So London, I'm going to have you tell me. What's in the middle that makes up the rungs of the ladder? Which part of the nucleotides are in the middle, making up the rungs? So like so if we had like a strand of DNA, I'm trying to draw, but it's like not letting me do it. Okay. So if we had a strand of DNA, this would be like the backbone of our DNA. The rungs are these in the middle, right? So the thing that it's like, we would draw a ladder like this, but there's 2 things in the middle, right? It's not technically a true all the way across thing. So what are those things in the middle? Just those. So, right, so we can call them the bases. You're not wrong, okay? So we'll call them the bases. It's more specifically nitrogenous, right?
Those are the A, T, C, and G.
The A, T, C, and G. Right. So don't just remember them as being just the A, T, C, and G.
Right.
What are they, T, C, and G? How are they defined, right? So exactly. So that's good. You got it. You're getting there. And so when we start talking about nucleotides, this is where a lot of us get confused. A nucleotide and the bases themselves, not the same thing. Right. But when we start talking about DNA and what it's made up and what its parts are of, we usually just talk about it in terms of the bases, because that's the important part. That's what codes for the DNA. And so what's really going on in DNA is in this backbone that we have here with these nitrogenous base. I should just deleted the whole thing. But here we go.
Structure is sugar phosphate.
Sugar phosphate. Yes, sugar phosphate. Sugar phosphate. Sugar phosphate. And the same thing on the other side, but it's a little bit skewed. I had to draw this weird, and now I got off of a. Okay. PS. Okay. So you can see here, if we started with a sugar at one end, start. We end with a phosphate at the other end. We start with the sugar at this end. We end with the phosphate at this end. This is important because we're going to get into what the structure of the DNA is. And this is the backbone of our ladder. Coming off of the sugar is whatever your base is, your A, T, C, or G. And these bases are hydrogen bonded to complementary bases. Okay, and then this, in this structure, this, why are you doing that? Sorry, this is a new tablet thing, and so it's a learning curve for me. Okay, so this is a nucleotide. We'll put NT for nucleotide. Sugar, base, and phosphate. So those are the building blocks. So there's one building block. Here's another building block. Here's another building block. Here's another building block. Each of these is a nucleotide. Whoops. I wish there was an undo. Okay. See how they're kind of oriented in opposite directions. That's important. We'll get to it in a little bit. So that's a nucleotide. And London got that one. Good. Okay. So I'm going to keep this up for our next question. So we have this kind of to refer to because we're going to get in a little bit more in depth in it as we go. Do your best to try to read through it. Okay. And DNA anti-parallel strands of nucleotides are linked by how are the two strands? So when we look at DNA, that is two strands. This is a double-stranded DNA molecule. All right, so that's not one strand, that's two strands. And how are they held together? Obviously by the hydrogen bonds that are linking those base pairs together. So our next person is Deontay. What are the two pairings that you can get in DNA, like the bases? How do they pair?
ATGC.
All right, exactly what I was getting at. All right, so what his answer was, when we have the bases that are pairing in there, A always binds to T. We'll just put some dots there to show that they would be hydrogen bonded, and C always bonds to G. I'm not going to ask you about which one's a pyrimidine or which one's a purine and how many hydrogen bonds they make, because one makes two and the other one makes three. I don't care, and I'm not going to ask you about it. Okay, I know you learned about it in chemistry, and yes, it is relevant, but I just feel like it's already a lot. So we're going to leave those. So A always binds with T and C always binds with G. Excellent. That's what's going on with the B's there. Okay, it's either A, and then the little dots, the A, if it's an A, it's going to go to a T. If it's a C, it's going to go to a G. Always. All righty. Next question. You know, we're going to jump into the complicated ones pretty quickly here in a second. All right, true or false, DNA polymerase builds DNA in the 5' to 3' direction. All right, so this one is true. Cool, right? And like, what does that mean? Like, I feel like that's the bigger question, really. Okay, so when we have that start, when we're talking about start, this is the 5', and this is the 3', and it's always that way. So on the other side, this is the 5', and this is the 3'. So one strand is running one way, and the other strand is running the other way. This is a property of DNA that we call anti-parallel. And they're anti-parallel because the backbones are actually running in a physical, like chemistry, the physical chemistry of that is running in different directions, okay? What's really going on there? I am not going to draw it out for you because it's a lot of like drawing the sugars and the phosphates and the bases and all that. What's really going on is the carbons of each sugar, and you guys remember this from chemistry, right? We're going to number the carbons based on like which one's bound to a thing and whatever. And the number of the carbon at whatever end of our molecule is where those numbers come from. So for the sugar molecule, the sugar in this is what I'm talking about here, okay? So if the 5th carbon is open at the end, that's the 5' end. If the 5th carbon is at the end, then it's the 5th, and if the 3rd, then it's the 3'. And that really is what it is. Again, I'm not going to show you a structure where that is drawn that you have to like in the structure, show me where the 5' is, or show me where the 3' is. But that's what that really means. And if you're just sitting here like, I'm not gonna, I don't care, good luck. I'm just saying that, okay? So, because this, if you're, that's just a fact that you're holding in your head and you're not actually relating it to how the structure is working. Like, I feel like that's where people get lost, okay? And you don't have to see the whole structure and the sugar and all those molecules in your mind. I'm not asking for that. But understand that those are like just different carbons in the molecule. And we're talking about the orientation of the molecules there. And that's what this discussion is about. So DNA polymerase builds DNA in the 5' to 3' direction. So what that means is if you have DNA that looks like this, you guys should, this should look familiar if you actually worked on your concept maps, right? If this is your DNA and here's your ladder and your A's to T's and G's and C's are bound like this, we will open up the strand during replication. This spot where we're opening it up, and we'll open it up as we go in further into it, this is called the replication fork. Replication fork. Something else that I want to be very clear about, and I will try to write it up here on the board, okay? Replication is the term that we use to just make copies of DNA. Only copy DNA. That's what it means. That's it. Transcription, also in this chapter, it's not a different chapter. Transcription is DNA to RNA. What if we were going from RNA to DNA? Anybody know what that's called?
Reverse transcriptase.
There, reverse transcription. The enzyme that allows us to do that is reverse transcriptase, which we will talk about in our activity, okay? And that is related to HIV, which is probably one of the most important of all of the pathogens that we'll talk about in class overall, which is why we spend a lot of time coming back to it a lot. Because it's not only important as far as being a pathogen that is significant in the human race, but also because it's taught us a lot about genetic information, how genetic information works. It taught us about viruses. It's not the only virus that can go backwards from RNA to DNA. What I'm telling you, what I'm going to remind you of, what we're going to talk about in our activity in a little bit, is that our body cannot take RNA and return it back to DNA. That is an impossibility. So when you get the RNA vaccine for COVID, does it affect your DNA? Please, okay? My mom did that to me over the weekend. She called me, we were having one of these discussions about the climate, as we're all feeling the political climate right now. And she was talking to me about, she said, well, but you know, we're having that issue where COVID vaccines are changing our DNA. It's just not worth taking the risk. And you guys, I get to look at you like you're stupid if you say that to me. And my mom has never taken a biology class in her life. But at the same time, I want to be very clear. It's impossible. Even if you have HIV or hepatitis, things that do that, it's impossible for that random RNA to get turned into DNA. With HIV, there are specific things that get involved that target specific genes and how they work and how they turn the specific mRNA into DNA and incorporate it into your own DNA. And reverse transcriptase is not the only thing it needs. It needs multiple factors in order for that to work. So it's not possible, okay? Just, it isn't, like this is not a discussion, it's just literally not possible. All right. Oh, another thing that I want to talk about really quick, because this was another thing my mom brought up, and I think that there's a lot of misconception about it. She asked me about like the risk for heart disease associated with the COVID vaccine, which I think we had mentioned briefly in the beginning of the semester because this was a common concern. You know, this is the thing. The statement is that the COVID vaccine causes heart disease, particularly coronary artery disease that can lead to heart attacks. There does seem to be some correlation, and nobody has proved the causation yet, but we have to prove causation before we can say it actually does. And does it or doesn't it? Nobody actually knows right now. That's the facts, okay? I'm not saying that it doesn't, and it might. But shall we talk about the risk for heart disease if you get COVID? That is proven, and it is significant. If you get COVID, the risk for heart disease skyrockets if you get COVID. if you get the vaccine, there might be a risk. I want to be very clear about that. And if you're like, well, if I get the vaccine and I don't get COVID, I will give you all my hopes and prayers that you're lucky enough not to get COVID. Whether or not you had the vaccine, how many people in here have had COVID for sure? Me too. Good luck. That's what I say to people that don't want to get the vaccine. Also, I want to be very clear about something else, something that has been shown and has been backed up by data. If you get this third booster, which many of us haven't gotten probably, just saying, because we're all like, oh, the vaccine is bad, okay? If you get the third booster for the COVID vaccine, that risk for the coronary artery disease goes down. So, just saying, maybe get your third booster, then you don't have to worry about it. More vaccines, not less vaccines. Okay?
So get the third vaccine, booster, does it need to be the same? that you
It's
got?
the same one. You have to get the same one that you got before. Yeah. Like if you got Moderna in your first two, you need to get the Moderna for your third one. Right? Okay. They just work the same way in your body. That way your body's getting the same antigen exposure and the same training. Otherwise, you do run risk of more side effects if you get the different vaccine. Okay. Yes. All righty. Any questions about that stuff? All right. So back to this question. We got to where DNA polymerase builds DNA in the 5' to 3' direction. I drew you our strand and what 5' to 3' means there. We've talked about how A binds to T and C binds to G. And now we're going to get into replication, copying the DNA, right? Transcription is going from DNA to RNA, and translation, exactly. Translation is RNA to protein. So I will point out something. Replication is not related to transcription and translation. They're different processes. And so when we learn about mitosis, which is one of the things we learned about in chapter 5, right? In mitosis, the first thing we have to do is copy our DNA. Then we have the copies, and then we line it up in the middle and pull it apart, right? So that required us replication. Whenever bacteria are multiplying, that requires replication. Now, will those processes also probably need transcription and translation? Yes, because anytime that you're making a new protein in the body, you have to go through transcription and translation. But they are different. They both are involving DNA, but it's how we're using the DNA. Are we copying the DNA, or are we taking information from the DNA and turning it into the product that it talks about? There's a lot of people getting confused between transcription and translation, or like which is which and how they relate to one another really. And so we'll get into that today as well. My best example is when we're cooking, like if you were cooking, and I'm going to give you my explanation when we get to that. It's not pertinent yet, quite yet, okay? But as we're opening up, what we do with our DNA in order to make the new copies for replication is we separate the strands and we make a copy of an old strand. And so what it would look like is while we're doing all of this, this will become, here's an old strand, and then here's an old strand. Let me get my colors. Hold on, because this is one of those drawings that definitely needs colors. All right, so bear with me. And then there'll be a new strand and a new strand. And so that's what's really happening up there. I'm not going to draw all the pink ladders, but you get the idea. So whenever you go from one strand to two new strands, the new strands are half old and half new. All right, there's a term for that. And we're going to get to it in the multiple choice in a second, but it's Catherine here today, Catherine Fields. Okay, Catherine, do you know what the term is? It is one of the major terms in the chapter. So I'll let you look it up. Okay. So when we're going from, we're doing replication, and our new strands are half old and half new. This is the process, we called it a specific kind of replication. And that's the term I'm looking for. DNA replication is blah, blah, blah. Okay, so I'll let you click on that. So over here on the left side, we can see that the strands are oriented in opposite directions. We call this term anti-parallel. This is because the backbones run in opposite directions. Okay? DNA replication. If you have two strands here, and again, they're still anti-parallel, all right? 5 and 3, 5 and 3. Again, referring to the carbons in the backbone. Right. So if you're going from, I kind of want to switch these. This is 3 up here. I wrote this. Y'all just let me write this, and you didn't even call me out for the way that I wrote it. 5 to 3. 3 to 5. Okay. So all this pink is throwing me off too. Okay. All righty. So when we start making our DNA, we start at the 5 and we add on the nucleotide that goes here. If this is A, then this one will be G. If this is C, then this one will be G. And we'll just add on these nucleotides as we go on, and we'll just add them in. And this is the 3 end of our new copy. Again, just like we're drawing down here what this becomes. This is the 5, and this is the 3, and this is the 5, and this is the 3. This was the 5 and 3, 5 and 3. Okay. Are you guys seeing this at least? This part, like the general, like big picture of it, right? All right, so this strand is made continuously. All right, and as we're opening up, so as that arrow goes in, that continuously made strand is pushing into that replication fork. We'll open it more and more and more and just keep making that strand. But we can only make, as this statement says, DNA polymerase can only build in the 5 to 3 direction. So what does this one start at? 3. So can we go this way? Can we just go straight in like that? No, that's a problem. Right? Can you see how that's a problem? You can't go straight in because why? Because DNA polymerase only goes in this direction, 5 to 3. This stopped using my thing. It only goes in that direction. So we can't go in that direction. So what do we do for the other strand? We have to go in the 5 to 3 direction. So in order to go from the 5 to 3 direction on the other strand, we will, this is the 5 here, and then if it goes into 3, we'll make it in parts. And as we open up that fork more, we can build another part. See how it's made in parts? It's made in fragments. Okay, so the fragmented one, all right, these fragments have a special name because science, okay? These fragments are called Okazaki fragments. All right. So the continuous strand is the leading strand. Dang it. The continuous one is the leading strand. And the one that's made in the fragments is the lagging strand. This happens every time that you need to make new DNA. You skinned your knee, you got to make new cells to heal it. You got to do this. Just like this, you have Okazaki fragments every single day, multiple times a day. This is happening constantly. This isn't some rare thing that you barely need to know about. Anytime any DNA is copied in any cell, whether it's eukaryote or bacteria, this is happening. Okay. All right. So this is how this is going. The lagging strand makes the Okazaki fragments because we'll make a fragment as it opens up a little bit. We'll make another fragment. As it opens up a little bit, we'll make another fragment. Okay. It's a problem because we'll have to make a lot of fragments. And these fragments don't just join up naturally. Special enzyme will come and join them back up together. DNA ligase. We're tracking? All right, so this is just DNA replication. We haven't even gotten into transcription and translation yet. This is chapter 6, and this is why I warn people, this is not the chapter to skip. Yes.
Like the replication work, do some people call it happen like unzipping
Yes.
the gene, and so DNA polymerase unzips. Is that the right thing?
So it doesn't unzip, helicase will unwind. And it unzips, like, it's a lot more complicated than that. Like, you guys don't have to know it. Like, I'm telling you, like, this is the baby version of this. Yes. It's a lot more complicated than that. Yes. But we can look at it as there's different types of DNA polymerase, and some of them are more involved in the unzipping, and some of them are more involved in the adding the nucleotides. That's why I don't want to get too broken down with like what does what and how. But for our purposes, helicase unwinds it. Because you guys all know that we don't just have a ladder. It's a twisted ladder, right? Yes, and it's twisted around histones. And so we usually think of the ladder as being like this. And like it's a helix, right? You guys know this would be twisted, right? But it's way more complicated than that. If I had a shoestring, I would show you. But you guys know you could twist the shoestring and that would be our helix, right? You can see the helix just kind of drawn like that is. But if you keep twisting it harder and harder, eventually they will start curling up on each other. You get the helix, and then the helix will start curling up on each other. That's what histones do. So it's a helix that gets curled up even further on itself, and even more. Not just like a phone cord. We're way beyond. If you keep twisting the phone cord and twisting the phone, I mean, maybe that's aging me a little bit, but you know what I mean, right? So you keep twisting it and twisting. If you keep twisting a cord more and more and more, eventually you get those weird little curly cues coming off of it. Your DNA takes that to like a level 10, does that like 10 times over and over and over. That's what the histones do. They stabilize that. And so histones are what keeps our DNA basically like tight and like not all over the place, okay? If you need to get in there and make copies or get in there and find a gene that you need to express, you have to unwind all of that first. And so helicase is the enzyme that helps with unwinding all of that, right? And then you have to open up your genes in order to make copies, whether you're making copies of the DNA to DNA or making copies of that gene in RNA form, you still have to open it up the same way. And so helicase is what is going to help us unwind and start opening up. And then the thing that's making the copy is either DNA polymerase, making DNA copy, or RNA polymerase if we are transcribing into RNA, okay? Yes.
So jumping ahead a little bit, how we open up later in the next chapter is by heating. This here will just be the heat.
The heat just forces it to separate.
Okay.
Yes, yes. So instead of using an enzyme to separate it, the heat will separate it. Yes. So
The same process.
the same process, same thing, yes, otherwise, no, for our purposes, okay? Okay, so this is replication. So did we find our term? Beautiful, beautiful, semi-conservative. So this is known as semi-conservative replication. Because each new daughter strand, this strand and this strand, consists of one old and one new strand. Perfect. All right. We'll get to some more questions here in a moment. This is a tough chapter. I want to give our foundation a little bit of help first. All right, so London's still not here. Our next person will be Taylor. We'll get to our question first. Okay. All right. Got all of our stuff drawn up here. I'm gonna leave it up here for a little bit, okay? Until we get into transcription stuff, I'm gonna leave it up. All right, which enzyme connects our Okazaki fragments formed during DNA replication? We do have that, we just covered it. It's in the bright green, but what was the name of that enzyme? So one thing that we'll notice about these are all real enzymes that are involved in genetics in our cell, right? One thing you'll notice about enzymes in general is that a lot of them have ACE at the end. So if you ever see something blobbity-ACE, like ATP synthase, for example, or something else, right? ACE always means it's an enzyme. Enzymes are proteins that help drive chemical reactions. And chapter 10, which is what we're going to talk about next Monday, chapter 10 is about enzymes and metabolism. And so we're gonna get into the enzymes and metabolism. And yes, we're gonna go over aerobic respiration again. Okay, so anytime though that you have ACE, those are proteins that act like catalysts. And we'll talk more about that in chapter 10, but they're helping drive chemical reactions, okay? So in this case, we're talking about DNA ligase, which is ligating, which means to join a thing. Ligating together our Okazaki fragments, so that's a continuous strand. If we didn't join our Okazaki fragments up there, then there would still be fragments. And then whenever we went in there to go find our gene to make a working copy of it, our RNA polymerase wouldn't know what to do with it. It'd be like, this is ended. I don't know what to do with this. So we have to join them, otherwise it's not functional. Okay. All right, our question for Taylor is, what does helicase do?
Helicase unwinds the genes.
That's good. Yeah, you're good. We'll just leave it at unwind. I don't want to get too complicated with this stuff. I want us to get the big picture idea of what all these things do, because it is a lot in this chapter, okay? So we want the big picture stuff. If you guys want to get into the nitty gritty of how this stuff, because it's cool stuff when you really start understanding it, but there is way more involved in this than what I'm teaching you guys. So yes, your question.
I don't think this will complicate it much more, but whenever helicase is unwinding, I understand that it basically straightens it out, but then doesn't it literally separate the two strands? Or what is the thing that makes them kind of
That makes
pull
them
apart?
separate?
Yeah.
So the tension that's being caused by the unwinding, as well as the binding of the DNA polymerase, not the one that adds the nucleotides, but there's other enzymes involved in that, cause the strands to separate. Okay, and it's just hydrogen bonds, right? It's just attraction. They're pretty easy to separate. Yeah, I don't say they want to unzip, but once we start adding that weird tension of the unwinding, they're already priming to want to separate a little bit. Now, here's the other thing. We have long chromosomes that are like millions of base pairs long, right? Each of our chromosomes are. And we're unwinding all of that. And now what I will tell you is, as we're unwinding and opening up our things to make copies of them, our ends kind of feel a little stressed from that, the ends of our chromosomes, because we're not in a big circle, we're in a line. And so what happens is, if you have shoelaces that are naturally wanting to curl, and we uncurl them in the middle, our ends will start, they're held at the ends, right? Our ends will start to hyper curl. And so we can't always make a copy of the ends of our chromosomes. The ends of your chromosomes are called telomeres. You've heard this, yes? So the ends of your chromosomes are called telomeres. They're just extra junk at the end of your chromosomes, because every time that we copy the chromosomes, we can't get quite all the way to the end. It's impossible because of the winding and unwinding involved. And so every time you make a copy of your genes, you'll lose a little bit of your telomere, and they get shorter and shorter. And you've probably heard about that too. As you age, your telomeres get shorter. At a certain point, you start losing genetic information, and that's where you might die. Like that's literally why we cannot live forever, unless we can learn a way to extend our telomeres, which is impossible so far. Okay. So every time, think about that, every time you skin your knee or something, and you're having to make new cells, those telomeres in those cells get shorter and shorter and shorter and shorter. And less often can you keep replicating them and making copies of them. And we think that that plays a big part in aging as well. You just can't keep replicating and maintaining that high quality DNA output for all of your genes in your entire chromosome. So that's happening as well. It is terrifying.
That's gonna like send me into an existential crisis. I'm so
Yes,
sorry.
and so there's a lot more involved in it than that, but that's the gist of it, and it is quite literally, you do lose telomeres, you lose genetic info the more you copy it.
Is that why in those like kind of bad movies about like non-aging and using other people for their DNA? Is that why they always use like kids in those movies?
Right.
Because it's fresh DNA.
So yeah, if I were to technically, if I were to know what your genetic information is and be able to clone you just from your genetic content, like build a new you from new A, T, C's, and G's, not like just taking your genes and forcing them to make a new organism, but literally like taking your code and building a new organism. I could build on telomeres. But what we typically do with cloning is that, like cloning the sheep Dolly, for example, we're taking a cell from Dolly, a sheep at some point, and making a new organism out of it. But that new organism that we make, it can only live so long because it's already got shortened chromosomes, because that organism already lived some. From the second you're born, you start copying your cells. you were just one cell. Yeah. And that cell that you were had the longest chromosomes that you ever had. Every cell that was made after that had shorter and shorter and shorter telomeres, short chromosomes. So you can't limit all of the loss of your telomeres. It's impossible. Just by growing, you lose some. But at the same time, if I'm taking the cells from an adult versus a cell from an infant, you're looking at significant difference in telomeres there. So where you want to clone is somewhere in the embryonic stage, and maybe the fertilized egg stage, maybe just a couple cells or something. Take that cell off and let it become its own embryo, and then be more likely to become a full-grown adult, for example. So that is an issue in cloning. We're talking about cloning multicellular organisms. Yes. Our bacteria can multiply and multiply and multiply, but they have circular chromosomes. They don't have telomeres. They don't lose any genetic information. They just go around the whole circle. So why don't we? Because we are way more complicated. We just don't work. Like our genetics are not set up to work that way. Okay. I mean, look at all the differences between us and a bacterium, right? We're not only multicellular, and all of our cells have to differentiate into different tissues. Like, think about it that way too. Each cell in your body, the cell that is in your eye versus the cell that's in your liver versus the cell that is in your toe, making up like the end of your toe that grows your toenails for you. I don't know, right? They all have the same genetic information in them, but they're getting signals from your body to only express certain genes, which is why you have an eye cell or a liver cell or a toe cell. Bacteria are just one thing. They're one cell. They don't have all that signal. They don't have all that differentiation. They don't have all that different function, and all that. So this is getting into all that and how that stuff is regulated a little bit. When we talk about operons, a lot of my students are like, man, that's so complicated. That's my introduction into how genes are regulated, because in our own genes, it's way more complicated. And you notice that we don't go into that a lot here. We will go into it in chapter 10 a little bit, but just like reminds you like, hey, we can regulate our genes too. But it gets so vastly complicated. That's a whole other course for a reason. Genetics is a whole other course for a reason. Okay. Cell and molecular biology, a whole other course for a reason. Okay. So we're just getting into the tip of the iceberg here. Any other questions before we go on? Okay, beautiful. All right, next question. Again, we won't be able to cover literally every single thing from this chapter, but I definitely want to get replication and transcription and translation. So during transcriptions, we're getting into transcription now. What's happening in transcription? This is a picture of transcription. Get rid of this. All right, so this is where I'm going to get into, actually, before I get into this, let me not forget to ask the questions for the day. John is our next victim here. All right, John. All right, so transcription is where RNA is produced. All right, so what is it called when we make proteins?
That's translation.
Right, yes, so I think it's up on the board and that's okay. But yes, that's translation. And so I'm going to talk a little bit about that. So I'm going to leave our picture of our replication fork up because I feel like it does kind of relate to transcription, which we saw in the picture. And I'll bring it in a second. I'm going to leave that little cheap note up there. Here. Does this look similar? It should, right? We're opening up our DNA, but instead of making copies of two sides, we're making a one-stranded copy of one side. And it's not staying bound to it. It's an RNA single strand copy of one side of our DNA. All right. One side of that DNA, we would, if we're making an RNA copy of a gene, one side of it we call the coding strand, and the other one is the template strand. So in this picture here, this bottom strand, this bottom strand here is the template. It's literally serving as a template to make an RNA copy. So if there is on the template strand A, T, T, G, then our RNA would be, what would it be? U. I heard it. I heard it. I heard it. U. I did it on purpose because I was hoping that somebody would hear it. So we're making, this is our DNA template. Then our RNA copy will be UAAC, right? And so it's serving as a template. But if we look at the DNA that matched up with that, on the other side, it would be TAAC, right? But the RNA has the U. So the one that is the DNA strand that will match exactly what our RNA is. If we read it, it would read the same thing, just replace the U's with T's. That strand we call the coding strand. It says the same code as our RNA will, but it's not actually serving as our working copy. So here's the thing. I am coming up here, or I have a whole big cookbook, right? Now, whenever you go to make a recipe, let's say you want to make, I don't know, spaghetti and meatballs, my dog's names. Anyways, so you want to make spaghetti and meatballs. You have this big cookbook and it has everything in it: burgers, spaghetti and meatballs, fettuccine, all this stuff in there. You can possibly think of pho, and it's just everything in this big old cookbook. Okay, and then maybe even that cookbook has recipes for everything that you could ever want to eat in your whole life. Okay, and all you want to make today is spaghetti and meatballs. Are you going to take that whole big giant cookbook over to your table and then make your meal? Probably not. You're probably going to open it up to spaghetti and meatballs and copy down the recipe for spaghetti and meatballs, right? That's transcription. So you opened up the DNA to spaghetti and meatballs and you made a copy of that recipe. It's just a recipe. When you make a copy of spaghetti and meatball recipe, did that make the spaghetti and meatballs for you when you did that? No, we ain't there yet. So transcription. And another thing that I think about this as, you guys know what a scribe is? Somebody that writes something down. Okay? Scribe needs to write. So we're going from DNA writing to still RNA writing. It's still writing, and the language that we have written it in is nucleotides, nucleic acids, RNA, DNA, nucleic acids. Yes.
So from the single strand RNA goes to a specific site for things to make, and it gives the base pair that needs to use to stay there.
So what'll happen is, let me think about it this way, because I'm not sure exactly. So how it happens is we're in the nucleus. DNA is always in the nucleus. Imagine you had that big fat cookbook and it sat in your pantry and you can't get it out of the pantry. It's like that. It's so big. You'll walk into the pantry and look for the recipe, copy down spaghetti and meatballs, and leave with your spaghetti and meatballs recipe, but leaving that big fat book in there because you can't get it out. So there's that. So transcription, anytime you're doing anything with the DNA, has to happen in the nucleus for eukaryotes. In bacteria, where does it happen? The cytoplasm. Excellent. So that'll be your points for today. Excellent. Happens in the cytoplasm because they don't have a nucleus. So quite literally, that is why. Okay. All right. Beautiful. All right. So we're tracking. So we have taken our DNA and made our RNA copy of it. And so the only difference between DNA and RNA, well, I say there's only a difference. Obviously, DNA is double-stranded and RNA is single-stranded in this case. But we're literally looking at the difference between our sugars, ribose versus deoxyribose, and the uracil versus thymine thing. Okay. But we've written down our copy. And think of it this way: your big cookbook was typed out by somebody. But when you go make your copy, you're probably writing it by hand. It's still nucleic acid. It's still the same language. It's just the typed compendium versus the working copy that you have. Okay. So that happens in the nucleus. Then we take that copy to our working area. In cooking, that would be to your kitchen, like your work table, wherever you're at, your countertop, whatever. In our cell, it's going to be the ribosome. Ribosomes are generally either going to be associated with the rough endoplasmic reticulum, or they can just be free in the cytoplasm, either way. But we have to get it out of the nucleus and into our working area. For bacteria, it's just cytoplasm to cytoplasm, which is unique for bacteria and also can lead to something else we'll talk about in a moment. Okay. So not only that, if we're in bacteria, and this is happening all in the cytoplasm, your ribosome, while you're making your RNA, you're writing your working copy, your ribosome is coming over there reading that. Okay, so we have to get the flour for the pasta. We have to get the eggs for the pasta. Okay, I see what you're writing over here. I'm gonna start working on it over here. In bacteria, that can all happen at one time. That's called co-transcriptional translation. We're translating the recipe while we're writing down the working copy. But in our cells, that can't happen that way. In our cells, we have to write down the full thing, take it out of the nucleus, and now translate it into our thing. So it's translation. So Spanish and English, right? If I'm speaking English and you want to translate it into Spanish, that's a different language, right? And it sounds stupid, but I'm gonna start there. Nucleic acids were our language for transcription. Okay, DNA to RNA to nucleic acid. But going from RNA to protein, are we still talking in nucleic acids anymore? No. What are our building blocks in protein? Amino acids. Excellent. So I'll count you off for that one. Isabella, right? Okay. So those are amino acids. They'll come together and build our proteins for us, right? And so that's a different language. So that's why we call it translation. It's also the big difference if we're talking about it being translation, different language. Now we're speaking in amino acids. That's where we're going to be going. Now we're not just having instructions and then a shorter working instruction. We now have the product that we wanted, which is the protein. Okay? Or your meatball and spaghetti that you're actually going to eat. Okay, so that's the difference. So to help a little bit with that, I'm hoping that it does. Okay, so good, beautiful. So that's transcription. And I mean, it is very similar to replication. It's just we're making RNA, and RNA is just single-stranded. Still happens in the nucleus. We're just using different nucleotides. We're using RNA nucleotides instead, right? It really is just the same other than that. All right. Right. A codon. What is a codon? 3 bases of what? Okay. So if I have DNA, here's my DNA strand, and it was double-stranded, and it was A, T, G, C, C, B, I don't know, G, who cares? And then our other side of our strand was T, A, C, G, G, C. All right. These 3 right here, in DNA, we call this a triplet. Oh my gosh, this is killing me, triplet. And then what we would do is separate that out. But honestly, I wish I'd give myself more space. We'll keep DNA in green, okay? We will separate that out to T, A, C, G, G, C. This will be our template. And then in RNA, to make the RNA version of that would be A, U, G, C, C, G, right? All right, this, and then if we would just get from that, while we're after making that, our RNA. This is killing me. Every time I accidentally click this button, I'm going to hold this pen differently. It's a learning curve. RNA. So then our RNA strand would be A, U, G, C, C, G. And they're all on one line, but I'm spacing it out so we can see the 3s, okay? This is a codon. This would be codon 1. Yeah. This is codon 2 in our RNA. All right? Those are codons. Codons code for an amino acid. So that's why they're called codons, because they're coding for an amino acid, okay? So AUG is our start codon. It codes for the amino acid, methionine, which I'm not gonna make you memorize the code, okay? You don't need to memorize the code. But familiarize yourself with how it works. So it just happens to be we always start with AUG, which also always it codes for methionine, okay? So our first amino acid, when we read this, would be methionine. But how does that match up? How do we get methionine from all this? What actually happens, don't you love it, is somewhere we have a UAC attached to, we'll draw him real fancy here, okay? Yeah, he's fine. He's not my best work, okay? But he looks kind of like this, and attached to him is the amino acid methionine. And I'm writing methionine like that. What does methionine actually look like? Remember, we've got the central carbon, an amino group, a carboxyl group, and coming off that central carbon is our R group. That's an amino acid. The R group, whatever that R group is, I'm not going to draw methionine. Okay? One, I don't remember how to draw them. Number two, I don't expect you to remember what they're all shaped like either. Okay. But that R group that is attached to that whole thing determines whether it's methionine, alanine, isoleucine, leucine, glutamate, aspartate, whatever. Okay. That's all that based on that R group. But that's what's really actually happening there. So this, what's actually, let me call on somebody. This one. Do we have, Jeremy's not here still. Do we have Kaylee Jackson? Do we have Jimena? Okay. What is that orange structure that I just drew? I guess it's yellowish orange. Do you know what that is in the process? So it's got a UAC in it, so I'll help you out a little bit. It is an RNA. So it's not mRNA, which is what our pink is. Our orange is a different kind of RNA. We only have to know 3 types of RNA. Let her, well, okay, I'll let you answer. It's okay. Huh? Yes, okay, yes, okay, good, good, good. Okay, so this is transfer RNA, and this is also known as tRNA. Either way would be a good answer. tRNA, also known as transfer RNA, because it's transferring that amino acid for us. And this is all happening, of course, in a much bigger structure. So, what was your name? Jordan, okay. So I bet you know what the name of the structure, what structure is this happening inside of? It's like a bigger thing, and it's made out of the other kind of RNA. The RNA, and I'll accept that, the ribosome, right? So this is all happening inside of, just give me blue, dude. Like, I don't know, I have to make this so difficult. This is all happening inside of a ribosome. And so one ribosome clamps onto this RNA. The tRNA comes into a pocket in the RNA, into the ribosome, and it's read there. We have another one that comes into another pocket that'll read the next one, and then it'll merge, it'll link together the methionine to whatever CG codes for, I don't know, leucine.
P site.
The P site and the A site and the E site, right? So the P site is where we make the peptide bond. The A site is like where the next amino acid comes in. The E site is where you exit once we're done with you. Okay, the whole thing merges. There is a video that is posted on your concept map page. There's several videos that show all these processes, and I highly recommend watching the videos because me drawing it is just a mess. But you seeing videos will make more sense. Okay. All right. Beautiful. Okay, so we got Jordan. Stallman. Good. Okay, we're making progress. We're getting there. All right. Chapter 8. So I'm spending more time on chapter 10 or whatever this is, 6, because chapter 8 kind of just feeds off of this. Okay. All right. Great. We're getting there. All right. So that process where the ribosome, which is made up of RNA, ribosomal RNA, is basically having the tRNA come into the pockets, and then that sequence, the UAC, Lauren's here today. Lauren's not here today. Okay. Kaya, yes, you're Kaya's, okay. All right, do you happen to know, in our tRNA, so we know that our codon here for our RNA is AUG. What's it called on the tRNA? Do you remember? I'll let you think about that. I'll watch it be the next question. I'll come back to you though, okay? So look that one up and we'll come back to that. All right. If it's the next question, then my bad. I don't have these all memorized. Oh, okay, it's not. Good, it's operons. We're just gonna completely just move on. Operons. In blank operons, proteins bind to operators unless a molecule that changes the repressor shape is present. So operons get a whole other level of explanation. Race. All right, so these are inducible operons. And I want to draw out the operons really quick, because operons, again, another thing that confuses people, but if we can just get the basic foundation down for our operons, it'll be a lot easier for you. Before we talk about the operons, did you happen to find out what that sequence is called on the tRNA? Exactly, anticodon, beautiful. So just the codon on the tRNA, that UAC is the anticodon. It's just the opposite to the codon. And because AUG binds up with UAC, we now know to bring in methionine. And that's just why we bring in methionine. So CCG would match up with GGC as the anticodon because GGC is on our tRNA. We now know to bring, I don't know what it's for, okay, leucine, let's just say, but you get the idea. All right, beautiful. Okay, so operons. How how confused do we already feel? If you made just your concept map, that might have been enough for you, as much as you wanted to have to deal with the codon. But you know you're going to get asked about it, okay? Or with a operon. So here's the thing: we have two types of operons: repressible and inducible. So I always like to start with inducible operons. Why? Inducible. No, I clicked it. I know I did. Inducible operon. When somebody is in labor, or well, okay, if somebody's pregnant and we have to induce them, that's us starting the process. They weren't having the baby, and now we're going to make them have the baby, right? So that's the idea with the inducible operon. Inducible, we can induce it because it wasn't happening. So they always start off and they can be turned on. That can be turned on, we would say that's inducing, okay? They can be induced. All right. The other one, known as repressible, it's the opposite. They always start on. You can't repress something. You can't turn it off unless it was on, okay? So start there always, because really you know what the words mean already. You know what it means to be induced. We turned it on. How can you turn it on? Because it was off. Just start there, okay? So the repressible is the opposite. The way all operons work is like this. Let me erase this because I want a little bit more space. I think we've seen it enough and we've made our notes already. The way all operons work, literally, is that you will have an operator, and then you will have a promoter, and then you'll have your genes, which I'll just put as gene 1, gene 2, and gene 3. It doesn't matter what they are, okay? But these are your genes. That's what the G's are, okay? O is operator. P is promoter. And then these are your genes. Then again, this is only in bacteria. Eukaryotes do not do this, okay? And that's really important to remember. Our genes are not regulated by one thing. Each gene is regulated individually, which actually makes it more complicated, okay? But for our bacteria, and then here's our genes. They're different genes, but they're all involved in the same process. So in this case, genes that help break down lactose, or genes that help make the amino acid arginine. Those were the two examples that the book gave. Those are different. One of them is an inducible, and one of them is a repressible operon. Okay, but the genes that help, they all work together to do the same process. All right, the promoter region is always the same thing. The promoter region is where RNA polymerase binds.
And to kind of just solidify this, that turns it on or off, and that's why all three genes work at the same time.
Right, so we're literally talking about, this is on the DNA, right? And the promoter is the region where RNA polymerase will bind and will separate the DNA and then make our RNA copy. That's what's going on, just to come back to transcription stuff, okay? So we already know transcription, now we got that out of the way. To make our RNA copy of the genes, that's what's happening, that's what we're regulating here. So that's where RNA polymerase would be able to bind. I drew this backwards, of course I did. I always start with the promoter, and I always stop to do this every single time that I do this. I'm so sorry. This is the operator and this is the promoter. It makes sense once I start drawing the things. Okay, so this is how they're all set up. If I could just get it right one time. Like I always start off the first class and I get it wrong. Okay, so what happens is we get something called a repressor. And repressor, I'm gonna draw it real big, binds on the operator. That's how all operons work, okay? That's where the repressor binds. RNA polymerase binds at the promoter, always. It doesn't matter if you are an operon or any other gene. That's RNA polymerase binds to the promoter and then makes the RNA of everything downstream. But at the operator, if a repressor is bound, you can imagine that it is physically blocking the RNA polymerase from moving down. That's what happens at the operator, okay? So it's like you are trying to drive somewhere and then there is somebody that comes out and stands in front of the road in front of you and you can't go now. It's quite like that, okay? So the repressor is physically blocking the RNA polymerase from moving. And that is how that will work in operons, regardless of what type you are. Now, if the repressor is bound, RNA polymerase can't move, the genes are off. So that's how inducible operons start. In a repressible operon, it will start with it open. And we're on, and we're making the genes. And in order to stop it, then that repressor will come and bind on and block it all, okay? So in a repressible one, it was on, and now we've triggered the repressor and it binds, and now we stopped making the genes. So with the lactose operon, the lac operon, which is an inducible operon, okay? You do need to know that it is an inducible one because I name it specifically in our learning objectives, okay? So in the lac operon, right, we start off like this. You are a bacteria and you come into media that has lactose, something you see every day. And you need to break down lactose in order to use it, okay? So what will happen is lactose will come in, bind onto the repressor, and then after lactose binds to that repressor, the repressor changes its shape, and then they both leave. And now we can turn on the genes, okay? That's how we turn it on. It's literally the opposite in the other version. So in the other version, like the arginine operon, a repressible operon, what happens is you have, please, why is it not letting me? Okay, that makes sense. You have a repressor somewhere floating out in the body or made by other genes or something like that, doesn't matter. And I'm not going to get into too much detail with it, but you're making your gene, you're making your genes, you're making your genes. So in the case of the arginine operon, what's happening is just like any other factory, we're making dresses, okay, summer dresses, and we're making them and making them and making them. As long as people are buying them, we'll keep making more. But suddenly now it's winter and people don't want a sundress anymore. And so now we're making them and making them and making them, but they're building up. And then the floor manager might say, okay, we need to stop making dresses. It doesn't make sense to keep making the dresses. And it's the same thing here. So what happens is, this is like the floor manager just waiting around watching things. Suddenly the body is making arginine, an amino acid, and it's starting to build up. The cell isn't using it. So it comes, the arginine binds with the repressor. And when that happens, now that whole mess can come and bind onto the operator and stop. So you see, they work the exact same way. It's just whether your repressor is bound at the beginning or it becomes bound to turn it off. Okay? So that is the best explanation that I can give you of an operon. Okay, so that's a operon. Beautiful. I know it's complicated, but trust me, you would thank me if I told you about not having to learn about how our genes work and how they're governed. Because it's a lot. It's a lot. So yes, this is all in bacteria. This is microbiology, right? So we're learning about bacteria. So beautiful. I'm not going to ask a question about operon because yeah, it's usually people have to work through that one on their own and make sense of it before we start getting too much into questions with it. All right, next question: missense mutations. And I will ask another question about another kind of mutation, so be prepared for that. Is Sammy here today? No, Sammy? This person dropped the class. Danielle. Okay, I will ask you about another type of mutation in a little bit. Do missense. There are several different types of mutations. Missense is one of them. In a missense mutation, we're changing the nucleic acid sequence that also results in the amino acid being changed. Okay, so Danielle, what is a nonsense mutation? To a stop. So let's say you were, UAA, and that coded for phenylalanine, but now we change it to UAG, and that's a stop codon. That would be a nonsense mutation. So if we went from AAA, which codes for phenylalanine, and then we changed that to UAA, which codes for leucine, then now that's a different amino acid. That's missense. Some of them code for the same thing. We went from AAA to AAC, they might still code for phenylalanine. If it did that, that's called a silent mutation. Still a mutation, nucleic acid sequence changed. But because we still code for the same amino acid, not as big of a deal. Okay. Yes.
So could a missense mutation also be considered a single polynucleotide polymorphism?
Exactly. Yes. So yes, if we had a missense mutation, and that's exactly what we see in the example that it gives in the lecture. If you watch the lecture for chapter 8, it talks about a single nucleotide polymorphism that leads to thrombosis, which is clot forming. So the normal nucleotide leads to a normal amino acid, normal clotting. But we had one nucleotide change that was a different amino acid, and we had excess clotting, which then can lead to high risk for stroke and things like that. Yes. So that was a single nucleotide polymorphism. Single nucleotide polymorphism is a fancy word, SNP. It's single nucleotide polymorphism. Poly is many and morph is versions. And all that means is that our genes can have different versions of themselves based on one nucleotide change. Sometimes that's good, and sometimes that's bad. Sometimes that can be deadly. Yeah.
Is that how pyrons form?
We don't think so. So we think that the prion is literally like normal amino acid sequence. And it's a healthy amino acid sequence. It's just
Is it just
teaching it
the
to fold a
structure?
different way. Yeah.
Okay.
Mhm. All right, beautiful. Okay, we'll get to they're all drawings. Let's do our little exercise real quick for this one, which is HIV. So it's going to be a little bit different. So, just get ready for the next one. Okay. I keep trying to hit a different button than what I'm meaning to. Okay, so chapter 6. Here we have HIV reverse transcription. When I wear a nose ring that has to face a certain way and it keeps rotating up into my nose. Okay. I'm sorry. You know them, but it's just very annoying. The things we do to be cute, you know what I mean? All right, so it's been 40 years since the discovery of reverse transcriptase. Reverse transcriptase, and we could read this. This is just giving us the idea, the primer idea of how HIV works. I'm going to make some notes out to the side to talk about HIV to go with this, but you're welcome to read through it if you would like to. Okay, but the idea is HIV, human immunodeficiency virus. Okay, it is a virus that has a positive sense single-stranded RNA genome. So this is getting into our questions, okay? Right, so that's our first question. Provided study discusses HIV virus. These HIV virus particles have a single-stranded positive sense RNA genome. So what this means is the ribosome can read it and make protein directly. So now we have a better context of what that means. We brought it up in the viruses in unit 1, but now we have a better context of what that really means. A positive single-strand RNA virus is the same thing as an mRNA. Our ribosomes can just read it and make the protein that it codes for. Okay? If it was negative sense, it means that we would have to make a copy of the complement of it. So if it was negative sense, if it said AAA, we'd have to turn it into UUU, and then we could read it. So that's what negative sense is. Okay? So this is positive sense RNA. So we can just treat it like mRNA. We can just read it and make the proteins. Okay?
Is that gonna function the same if it was a DNA strand rather than an RNA strand?
No. So That's the idea there. So when you have DNA, always when you have DNA, you will have your two strands and you'll have to turn one of those strands into a single strand RNA. A single strand positive sense. And then your ribosome can read that and make a protein. And this process here from going DNA to RNA, this is transcription, right? And the process of RNA to protein is called translation. So we'll keep that at the side for us to refer to so we can recall that stuff, okay? Erase this a little so we can see better.
So if you're a virus and you have DNA instead of RNA, it's pretty much you have to go through a few more
Transcription,
extra steps
yeah.
before you can infect.
Exactly. And DNA viruses, because they're DNA and all the stuff that we do with DNA happens inside of a nucleus, DNA viruses have to get into the nucleus. Yeah. And then they do all of their DNA stuff in the nucleus. Now, to make it clear, phages. You guys remember bacteriophages, viruses that infect bacteria. Bacteria don't have a nucleus. And bacteriophages are all DNA viruses. Well, typically, okay? But they don't need a nucleus, so it's a little less.
So it's a little bit
complicated.
easier for.
It's a little easier for them, yeah. Okay, so, right, so this is our general idea. So HIV is a positive sense RNA genome. We can just start making the proteins to build new viruses right away. And we can even use that single strand RNA genome to make as a template to make new RNAs if we wanted to, right? And you know, the virus can dictate that. But this actually with HIV, significantly more complicated than that, because it won't just stay as an RNA genome. HIV will use a special enzyme and turn that RNA genome into double-stranded DNA. Which is a copy of that genome that's double-stranded DNA. And then we'll insert that into the host DNA genome. Right? Once that is inserted into the host DNA genome, we now call it, that beautiful term that we were so frustrated about in the test, a provirus.
So it's all about the retrovirus and we go down.
So yes, anytime that we have a viral genome that's now incorporated into the host genome, we call that a provirus. And we're like, just like we talked about with prokaryotes, before a nucleus, this is before a virus. It's not the virus anymore, right? It's the virus's genome that's incorporated in the host genes, kind of becoming integrated in the host genes. It's a provirus.
And then the prophage is just a trait that was gained.
No, so prophage is whenever we have this same process happening in bacteria.
Oh,
And then when we get a new trait from that, whether that's the lysogenic conversion,
Yes.
beautiful, beautiful. Okay, so we're bringing it all together. Okay, so we're just getting into more detail with that here. This process of going from single strand RNA to DNA, this is biology and how we know our cells work. DNA, we make an RNA copy of it, and then we use that RNA copy to make our protein, transcription and translation, right? That's the order of events. Going from RNA to DNA, we talked about that previously at the beginning of the class, but if you missed it, that process is called reverse transcription. It's just like transcription, DNA to RNA, but the reverse. Reverse transcription. And the enzyme that does that is reverse transcriptase. So this is a special enzyme encoded by HIV and some other viruses, retroviruses primarily, but there are other ones that can do it as well that are very similar. Okay? We can get really complicated. And I can tell you guys about hepatitis B. Hepatitis B is a DNA virus that encodes reverse transcriptase. So that gets wild because it's the DNA virus. So what it does is it goes through DNA transcription. It makes single strand RNA so it can make the protein parts, but then it uses the single strand RNA as the version to make a new DNA that gets inserted. It just has an extra, I don't know why, but that's just how the virus works. Yes.
And the hepatitis B is the one that is passed on to infants through birth.
Yes, Hep B, and we can see that happen with Hep C too, but that's the more common one that's associated with that. Hep C usually needs direct blood transmission, so it's more like intravenous drug use. Hep C. Hep B is like incidental blood contact as well as sexual contact. Hep B is. Hep B is more commonly associated with cancer, but both Hep B and C can cause cancer. Once you have Hep B, you cannot be cured of it. It's sort of like HIV. For this same exact reason, it gets incorporated into your DNA. And how do you remove it after that? Hep C can be treated. Once you're at certain stages of Hep C, it can be treated. There's antivirals that can actually treat that. But we have a vaccine for Hep B. We don't have a vaccine for Hep C. Yeah, for now. Yes, for now. We probably will at some point, but yes. So that's things to remember for the future, okay? But that's not on this test, just to be clear, just to put this in perspective, we're learning about it in the context of real life scenarios, but FYI for unit 4, that is stuff you do need details like that. But anyways, HIV has a positive sense RNA genome. That's its normal genome. It doesn't come with DNA. So it turns its genome into DNA through the reverse transcriptase enzyme, okay? So that is our questions 1 and 2 out of the way. The study mentions HIV produces reverse transcriptase. It allows the virus to, do that, to make to take its RNA genome and turn it into DNA. All right, so then our third question, what role would DNA replication play in our HIV viral life cycle? So it will play a role. Even though it's a RNA genome, once it turns itself into DNA and inserts it into your genes, your cells will replicate in their own DNA as well as also replicating the HIV provirus. Okay. So anytime that your cell replicates itself, it's also replicating the provirus. We tracking with that? Okay.
Also replicate the offspring too, that's a thing.
So not unless it infects the gametes, right? So as it turns out, HIV only infects T cells. And I do not want to get too much into that now because that is unit 3 stuff, and we haven't learned about it too much, but they're one of our immune cells, right? So it only affects CD4 positive T cells, which are captains. They like govern everything your immune response does, which is why it leads to immunodeficiency. If you have a virus that infects your immune captains and then you kill your immune captains, you can't govern any immune response anymore. Which is exactly what happens with HIV, okay? But your immune cells don't get passed on to your offspring genetically, right? So HIV doesn't get passed on at the genetic level that way. But can you transmit the virus to the baby and infect the baby? And then yes, that can happen, but it's not like genetic transmission of the virus, okay? Yes, all right, so question 4, the study mentions that reverse transcriptase is one of the most common targets of antivirals for treatment of HIV. I mean, it is by far that between reverse transcriptase and another enzyme that HIV has, which we'll learn about in unit 4, integrase. So you have to have both of those. Reverse transcriptase just turns it into DNA. Integrase integrates it into the host genome. You have to have both there, okay? So both of those are easy targets for antivirals. So why would they be easy targets? Because our body doesn't have reverse transcriptase or integrase. So those are just like peptidoglycan was a good target. This is a great target for an antiviral. Means that there's less toxicity associated with it. Yes, so most of PrEP and STEP in preventing HIV, as well as our HIV treatment medications, inhibit reverse transcriptase. So we can't keep spreading it to our other T cells in our body. Once it's incorporated in your T cell, that T cell has that forever. We can't get it out, but we can keep new viruses from being made and keep new viruses from infecting more of your T cells. Are we tracking a little bit? Any questions? If reverse transcriptase can convert RNA into DNA, how might this be used in research and medical advancement? RNA viruses that we want to work with at a genetic level and manipulate and learn more about, we can use reverse transcriptase to turn them into DNA. DNA, you might not think about this that much, but I tell you, every scientist that ever works with DNA or RNA will tell you, are very difficult. Work with DNA, it's bigger, it's easier to manage, and we can use it in PCR to make copies and things like that. You can't do that with RNA. RNA is single-stranded. How do you make copies of RNA? You turn it into DNA, and then you make copies of that, and then you can work with it. So this has radically changed the scientific industry. The ability to now work with RNA and turn it into DNA, which is much more stable.
Could you also use that for like tracking epidemics over time? Because I would assume it's kind of like a specialized thing that you can use.
Yeah,
to,
so like looking at the difference between like a modern day HIV reverse transcriptase, which is commonly used as a model, which is semi-immunodeficiency. You can like compare, yes, the genetic differences between the reverse transcriptases of those
Like
guys.
you can use it not to like say that it's mitochondrial DNA, but you
It's
can use
similar,
it
yes.
almost.
That unique parts of those viruses, whatever it may be for whatever virus, used as a comparison method, tracking them over time, genetic mutations, evolution, and stuff like that. And there's other, parts to the viruses that we might use that are better predictors, but yes.
So we use HIV nowadays, right, to put it into people's cells? Yeah.
Yes, so that's our next chapter. That's why I wanted to talk a little bit about HIV, because we can actually hollow out HIV, remove all of its bad guy crap in it, and then use it to insert new genes into your body. And that is gene therapy. Most gene therapy that is used today uses HIV. And so we use HIV, infect your bone marrow cells with HIV, but instead of it, and you can make your own version of that protein forever and always, and it's built into your.
Technically HIV positive?
No, because it's not really HIV. It's like, we just use it as a shuttle.
You're,
We
yeah,
hollow it
you're
out.
just using like the name of it.
Uses the
It uses
gene
like
that
this.
you needed instead of the HIV crap. Yeah. All right, beautiful. So we'll talk about gene therapy. That's in the next chapter. No, it's good. This is good stuff. And I feel like this stuff is like, talk about like a horrible disease that it's one of two diseases that we consider to be 100% fatal. Not many diseases we can say that about. And now with HIV, we have treatments that can prevent it from killing you, right? But if we don't treat it, you will die from HIV. Okay? It's 100% fatal if you don't treat it. There's other diseases out there that is 100% fatal if we don't treat it. And there's really no treatment for it, which is rabies, which we've talked about previously. But so there's that disease, one of two diseases that is 100% fatal if it's untreated. And it's wildly common across the world. Half of South Africa has HIV. Half of them. And this is a permanent, like you have it forever. Once you have it, you are HIV positive forever. Even if your viral loads are undetectable, you still have the ability to make the virus if you come off of your medications. You're always HIV positive once you have it. And so it's a lifelong disease, and it's a battle, and it's something that people that are positive for HIV will constantly have to battle unless we can develop a way to remove the genes from your body, which they're working on it, but we're nowhere near that right now. So for now, it's a lifelong infection. But the advances that have come, the life-saving advances, I can't even touch on the importance of them, that have come from discovering and understanding HIV. I mean, we're saving lives with the knowledge we got from HIV. So it's a double-edged sword. Like there's a good side and there's a horrible side. Like I don't know how else to say it. But it's like a lot of medicine, I guess, in that regard. But
It's like
a lot of medical history when we talk about that stuff. Like you had to get from the good,
a lot of drugs that
the good
I
from the
don't
bad.
know.
Yeah. All right. So sign in again real quick. We will make chapter 8 pretty quick. A lot of good discussion on chapter 6. Chapter 8 is more or less just finding ways that we can manipulate our understanding about the genetic code to our advantage. Let me see. London's still not here. We got him in it. Lauren's still not here. We got John. Just trying to clean this up while you guys are getting signed in. Sammy here. Just want to double check. I know that she wasn't here just a second ago. All right, we still got K.
Are they trying to do that with like any other viruses other than HIV? Like, are they
Yes,
trying-
there's other ones that they use for that as well. Yes. Some of them, yeah, we
But
look into
HIV
it.
is
Yeah.
not one they
Yeah.
use.
Yeah, there's other ones that they use for that too. All right, we will have Kay and then Jackie and then Amna. Those are our last 3 for today. Okay, just so you know. Is Kay here? You're Kay. Okay. Jackie's not here today, right? So then just you two. As long as we get you two out, we'll just kind of crank through the other ones. Oh, let's get going. I'll try to make you guys the easier questions if I can. All right, chapter 8, first question. Double check this thing and make sure that I'm not missing anybody. I'm not. What type of biological macromolecule are restriction endonucleases? All right, so I'm not going to ask a question about this one, but the reason they're restriction endonucleases, they do cut nucleic acids. So that's where we're probably pulling that from. Restriction endonucleases cut nucleic acids, they cut DNA, and they make the sticky ends. And they cut at palindromic sequences, which are things that can be read the same forward and backward, okay? But ACE, these are enzymes, and enzymes are proteins. So this is a protein that cuts. Just like we learned, DNA ligase is a protein that seals together fragments. This is a protein that makes fragments, cuts. Okay? So it's a restriction endonucleases. Beautiful. Question 2, restriction endonucleases, more about them. I mean, there's a picture. I feel like it's kind of a little bit of a giveaway.
Xerox machine, like a printer?
I thought that,
Like
that's what
my-
it said on the, in the book.
So we would say that PCR is like a
Oh,
Xerox
PCR.
machine. Yes, yeah. So restriction endonucleases, these are enzymes that basically like, let's see if I can. Really quick. If we have CAAT, every time I click that button, TG. All right, this is one DNA strand. Remember, we have 2 strands on DNA, right? What's the other strand read? Oh, actually, let me make a question. K. You want to tell me what the next strand? No, you don't have to tell me what's the next letter. What goes with C? Excellent. That's good enough. All right. G, T, T, A, C. Right? You notice how this reads the same as that. It's a palindrome. Right. Okay. So this is palindrome. That's what that means by palindrome. And what the enzyme does is it cuts like this. And then what you end up having is a free end. That free end of the AATTG is just free. And then the other end with the TTAA, whatever, that end will be free. These are sticky ends. And I can then make a gene that like, hey, people make insulin. And I'll put at the end of my insulin, the TTAA stuff, so that it'll stick in there and just insert. I cut my bacteria gene and then inserted a human gene using the sticky ends. This is how insulin is made, by the way. We literally cut a plasmid, put human insulin into it, and then sealed it up with DNA ligase. So you can't have a fragment. You still got to seal up with DNA ligase. And then put that plasmid into bacteria. Now bacteria make insulin, which is why it's important. It used to be we had to get it out of cattle or dogs or like animals. We had to get it out of animals and extract it from the animals. Now we can just force bacteria to just get out like mad. And they'll make that insulin more affordable. And so now more people who have diabetes survive than what they did back in the day. Used to be that if you had diabetes, you basically just died. It was a death sentence.
Didn't it like take off your fingers and toes?
A lot of amputations associated with that. All right, so that restriction endonucleases, they cut at palindromic sequences, and that's probably the next question. Who knows? Restriction enzymes can cut anywhere in the DNA sequence. So do they cut anywhere in the sequence, or do they cut at the sequence? Okay.
You gotta stop like answering these questions. So.
I know. It's all right. We're talking about it. We're putting it into context. We're having discussion, and then we're testing ourselves in a different way, which is something I feel like a lot of us, just more exposure to. It's fine. So obviously they can't cut anywhere in the sequence. They can only cut at these palindromic sequences. In fact, they can't cut at any palindromic sequence. There's specific ones that cut at specific sequences. So ECOR1, which comes from E. coli, the E. co part, R is restriction enzyme, and then 1 The number one that we picked from E. coli can cut at like this sequence. And then another one, EcoR2, might cut at a different palindromic sequence. And so we have different enzymes cut at very specific palindromic sequences. Many of them create the sticky ends. This is a better picture than what I could draw. See the little sticky ends there? So many of them create sticky ends, but some of them have blunt ends. So that's not a given that you get a sticky end. We can use them for different things in molecular biology. These are naturally made by bacteria, by the way. They're made as a defense mechanism to cut out like viral DNA and stuff like that. Or, you know, they took up a plasmid, but it's like a harmful plasmid. Now they can cut it up into pieces. Okay. Well, we used it in molecular biology for our own purposes, like again, to insert genes into things, as well as for molecular fingerprints to see who done it in a murder. Because when I cut my genes with EcoR1 and your genes with EcoR1, if I get your blood and I get my blood and we cut our genes and somebody was murdered here and we found some blood that seems to match the victim, but also some other blood.
But who's the victim?
And we could say that who's the victim or who's the perpetrator, but we can use this to cut up the DNA and see if the fingerprints match. If you've watched the lecture, this is in, I can't draw it, okay, but like the way that it matches up, that's what we do in like CSI and things like that, crime scene investigation. All right. Yes, those are called restriction fragment length polymorphisms, RFLPs. All right, all right, whatever, this is fine, good. All right, I don't know if I want to ask a question about this one, but Amna, you're coming next. All right, true or false, CRISPR-Cas9 is a protective system in bacteria that can be used in genetic engineering to cut just about any sequence. What do we know about CRISPR-Cas9? Is this true or false? Probably not going to ask a question about this one, Amna, but probably next one. All right, CRISPR-Cas9 is a system that bacteria have naturally that seems to be able to have, we can basically tell it, this is your blueprint sequence, this is your template sequence. If you find this sequence in a body, cut it out. And so it can literally be told where to cut things out. And so we might be able to hijack this to cut out DNA that's associated with the provirus for HIV, or we might be able to use it to cut out bad genes that code for something harmful like cancer or something like that. So it is in the works right now being investigated to be used in somatic gene therapy right now, and potentially maybe also germline, which is heritable, but it has not been successful in large multicellular organisms so far. In fact, so far it seems to be very hard to manage and regulate even in mice. So I don't, it'll happen, it will happen. We will be able to do stuff like that. But it's not there anywhere near there yet. Okay. If you ever watch, there's been some documentaries out, man, that some people try to do this in their garage. These are people that literally have no idea how genetics in your body works and how it regulates, and they just have a general, like if you learned it in my class today, you'd be like, oh, I can like cut out and add in genes to make, I can turn my body green by adding in green fluorescent protein. In theory, yes, but in the actual practice of it, if our best pharmaceutical companies can't get it to work. And so a lot of people be like, well, it's being kept from the pharmaceutical companies. They're just not letting us have this because it can heal us from everything. Bet if they could do that, they would be marketing it and getting all the money off of you that they could with this stuff. Bet, but they can't. They can't even do it. So unrealistic to think you can just do it in your garage.
Honestly, props to them for having the spirit to do it.
Right, and if you watch it, yeah. Well, yeah, anyways, PCR is a technique. What do we use it for? PCR. PCR works quite literally, exactly like how this picture is drawn. I'll show it in a second once we get our question stuff. We'll kind of go over it. All right, so real quick, Omna, what does PCR stand for? Yes. Polymerase chain reaction, that first word is the hard one, right? Polymerase, or yes, and poly means many. Merase just means an enzyme that makes many. Chain reaction because it's a chain reaction. So what happens is we start with a double-stranded DNA sequence. That's what this is. We split it apart with heat, and then we make a copy of that part. You have to use a primer. So one thing we didn't get into a lot of detail in our review of replication of is DNA polymerase can't just make a copy. It has to build off of something. It has to attach those nucleotides to something. And so in our DNA replication, it uses an RNA primer, and then it'll go back and replace it with DNA sequence. But in our PCR technique, we'll make the primers. And that's actually how we govern what PCR makes. So if you're getting tested for COVID with PCR, what actually has to happen is we have to have primers there that see COVID genes. So I can't just swab your nose, put it in a PCR machine and say, tell me what this is. It has to have primers for things, and those primers are very specific. Can you have a mixture of primers for COVID and primers for flu and primers for RSV? Yes, but we have to engineer it in a way that you can tell the difference of which one is being recognized. That's actually way more difficult than you know. All right, so most of the time, it's only going to test for one thing or another. Okay, but beautiful. All right, let's move on. Yes. Yes, in vivo is inside of the body. In vitro is like in a test tube outside the body. So if I put a swab in your nose and then I put it in a machine and it tests it, that's in vitro. Okay. In situ, it's usually using like tissue biopsies from a person. So it's kind of like from a living thing, but it's not still in the living thing. Okay, so there's some of that too. And in situ, I think it was talked about in this chapter. If it wasn't talked about in this chapter, it will be in our diagnostic chapter, which I think is in Unit 3. But yes, so nucleic acid hybridization, which was definitely talked about in this chapter, is usually done in situ. Okay. All right, we're gonna do one more question, maybe. Let's see, and then we'll do our activity real quick. Which of the following is not one of the stages in a PCR cycle? I learned PCR has 3 steps. One of these is not one of them. All right, so the answer for this one is the green one, ligation of the primers. We don't need to ligate the primer on, we're just building off of it. PCR literally is just going to tell you whether the thing is there or not. And that is it. So if I put COVID DNA into a PCR machine with primers for COVID, what happens is heat separates your patient sample DNA. Then annealing is the next step. So denature is the separation. The word for that separation is denature. Use that heat to do that. Heat. Would it also denature the polymerase that's going to be adding on the nucleotides? So we have to use a special polymerase that we got from bacteria that lives in like thermal vents or whatever, because we're going to heat it up to 92 degrees Celsius, which is nearly boiling, which would kill your own DNA polymerase. Okay. So what we do is we heat up the DNA to separate the strands. Then we add on our primers by cooling it a little bit, and the primers are in excess. Primers bind on. If they bind, that's because the gene is there. If they're just still floating around the milieu, they won't extend because the gene's not there. And that's literally it. You either have it or you don't. That's all PCR does. It's literally just making copies of the gene if it's there. If the gene isn't there, if I swab your nose and I put it in the COVID PCR, but you have the flu, nothing will happen. And that's a negative test. If you had the COVID gene, we'll copy it. And the machine can sense that, and then you're positive. Okay? That's literally it. Y'all thought PCR was fancy, and all we're doing is just making a copy, or it's not there. And that's it. Okay? You have to know what gene you're looking for. Okay, so we'll stop there. And we'll go really quickly over our gene therapy example. Go away. It's on this. Just really quickly. This talks about sickle cell disease, and we have effective gene therapy for sickle cell disease. We do. It exists. If you have sickle cell disease, which is a common trait, usually in people of African ancestry, okay? And what it means is that you have a malformation of the shape of your red blood cells. And it can also impede how the blood cells can carry oxygen. It can lead to clotting issues. It can lead to a lot of different issues in the body. Okay, if you have 2 versions of that gene, then you have a version of the sickle cell disease that is usually deadly, but But if you have just one version of the gene, then that can even lead to expression of misshapen red blood cells and expression of sickle cell disease as well. So it is a potentially deadly disease, but also a life-affecting disease if you have just one version of the gene. Okay? So we have found a way to use HIV virus to insert this gene into the bone marrow of these folks. And now the new red blood cells that they make have normal trait instead of sickle cell trait. Okay?
But would this one be inheritable?
It would not be heritable, correct? Yes, so this is somatic. So far, we haven't worked much with inheritable because you have to do inheritable changes at the embryonic or fetal level. And so I can see issues with that, right? So yes, so far it's not heritable, right? All right, so the questions that are associated with this. Gene therapy, the difference between somatic and germline. Somatic means we're changing like your body cells, not your gametes. We have to change the gametes for it to be heritable. Most of our gene therapy stuff has to deal with like blood cells, whether it's white or red, okay? And so that usually can do in your bone marrow. All of your blood cells come from the precursor in your bone marrow. So we can basically replace your bone marrow, okay? And so we can do that way, but that's not heritable. Your children won't have that gene, okay? Because your gametes don't come from your bone marrow cells. They come from your testes or your ovaries. We're not changing those. All right, what are some potential benefits of somatic gene therapy for people with sickle cell disease? Obviously, we're eliminating the disease and giving them normal red blood cell function, right? Considering the estimated cost of gene therapy exceeds $1.5 million per patient.
Yeah.
Now you're understanding why you ain't heard much about it. So
What are the
it exists. It is not FDA approved, right?
Well, I can.
Yes. Number one, the cost to treat a patient. Number two, enough patients that are willing to have their genetic information changed. Now, if I were to have told you before you walked in off the street and learned about microbiology and genetics, and I'm going to infect you with a modified HIV virus that's going to get rid of your sickle cell disease, y'all would probably be like, well, I'm good.
I'm good, actually.
I'm good, actually, yeah. So, but now that we've learned more about this and a little more information, now we might be like, why aren't people doing this? But they're not educated on what's going on here, especially when a majority of these people might be living in, again, if we have, aware of what is going on in our world, not just in third world countries, but here in America, how many of our people who are of Black ancestry are going to be living in low income situations, even in the US, right? I mean, let's be real here, okay? And how many of them are going to have their government-funded healthcare pay for gene therapy? Or go through this trial even, right? So that's an issue. So there's stuff that associated with that also feels like we have this thing that can treat you. The government's not going to pay for it though, and it's going to cost $1.5 million.
For like select.
Is that why it is so sensitive? Is because they're not gonna FDA approve it to lower it?
Who knows? There's a lot of stuff that goes into that stuff, right? And I don't know why some things are as expensive as they are. I'm being quite honest, just being real there. But yeah, so that's part of it. And I mean, it might become FDA approved. It might be able to be one of those things that might be a little bit more available. But at the same time, in order to treat somebody, we have to do a bone marrow transplant essentially. And so just everything that's involved in that, it's risky in and of itself. But this can kill people. Like having sickle cell disease can kill you and can lead to all sorts of effects of your body that can become compounding and lifelong. They can even have their spleen can rupture just because they have sickle cell disease and stuff like that. So anyways, how might previous success and failures in gene therapy trials, such as those involving some patients that are listed in the thing, you want to read it, okay, influence patient perceptions and decision-making about gene therapy in general. There's been people who have had negative effects. We used the gene therapy and they had a reaction to other aspects of the virus that we didn't know about. We're putting a virus in somebody and they might react to that virus and not just get the gene that they're benefiting from. Also, we're changing your genes. We don't know what other effects changing your genes can have on people. So we might have negative side effects. And whether or not those negative side effects were associated with that process, or like just having a bone marrow transplant and infusion and stuff like that, and those things might make you less likely to want to take the risk associated with that. So this is just weighing the risk. Same thing as what we face with vaccines today, right? Weighing the benefits and the risks. We got to weigh the benefits and the risks, but we got to be also educated about all sides of those benefits of the risks in order to make proper decisions. In what ways do ethical, legal, and social issues, known as ELSIs, extend beyond affordability? Obviously, the affordability is an issue here, but at the same time, we're talking about changing somebody's genetic makeup. We can change their sickle cell disease. That means I can change your eye color. I can change how tall you're going to be. I can change your hair color. I can make
Yeah.
you more likely to lose weight.
People would
And
find.
the people that can afford
Then
that.
we get the, you
Yes,
get.
then we get into eugenics, exactly. So that is clearly like, right here, we're talking about a life-saving procedure. But this is technology that can set us up for eugenics. And if you can afford to have a designer baby, then your baby will succeed in the world. And if I can't afford to have a designer baby, then my baby's gonna be cleaning up trash. You get what I'm saying? Just because you can't afford to design your baby. Has anybody in here seen the movie Gattaca? It's exactly that. If you haven't seen it, I recommend it to Ethan Hawke and Uma Thurman. And it's such a good movie. But that's the idea of it. Like, are you better than another person that has been designed? So that's where this gets from. I know that's a wild, like, it seems like it's so disconnected, but it's the exact same technology. So that's what we're getting at too with like CRISPR and stuff like that, like literally editing that stuff. Yeah, okay. So that's it for today. We'll head up to the lab and we're gonna be looking at pond water and stuff today. It should be cool stuff. It'll be fun. It'll be fun today. I know it sounds lame, but it is pretty cool. Yeah.
Honestly, you say that, but on TikTok, I keep seeing